So welcome everyone to our GT Neuro seminar. This is a really special one. We have it's co-sponsored by the GT and Emory Neural Engineering Center and the McCamish Parkinson's Disease Innovation Program. It's my pleasure to introduce our speaker today, Doug Weber. Doug is the Akhtar and Buddha Professor of Mechanical Engineering at the Neuroscience Institute in Mechanical Engineering and the Neuroscience Institute, at Carnegie Mellon University. He's also one of the external scientific advisory board members for our McCamish program. So welcome, Doug. So Doug got his bachelor's in biomedical engineering from Milwaukee School of Engineering, followed by master's, correct me if I'm wrong, right. Master's and PhD in bioengineering from ASU, from Arizona State University.
Go Devils.
There you go. And then he did his postdoctoral work at the University of Alberta in neuroscience. So he's kind of speaks to a lot of our language here across engineering and neuroscience. He then joined the University of Pittsburgh, where I first met Doug, many of us did, the bioengineering program and the neuroscience programs, where he started the Rehab Neural Engineering Laboratory. And I was delighted to see that his own description of his work was he's really focused on sensory feedback and sensing, even though I think of him as a movement control guy.
The closet sensory.
Yeah, closet sensory. Understanding the role of sensory feedback in supporting and regulating a wide range of perceptual, motor, cognitive, and autonomic functions. That's his language. I couldn't have said it better. We know him from his work in spinal cord stimulation and recording across many different animal models, including humans. So you may see smatterings of different things today. And in particular, focused on sensory feedback, and he's published and motor neuroprostheses. You'll hear about some of that today as well. He's published widely across lots of different avenues of the literature in engineering and neuroscience. He has he's done a lot of translational work, and he has 8 patents, maybe more by now, I don't know, and a very strong commitment to mentoring. So it's really exciting to have. I know there's a lot of students and postdocs in the audience, and importantly, he also, in the middle of all this, became a DARPA program officer and was the founding member of DARPA's Biological Technologies Office. And he created a really exciting program and had a wide-ranging portfolio of neurotechnology. And a lot of this was pushed on by the Brain Initiative program that started in 2013. So it was a really exciting time. He served his time, came back to civilian life.
Service, not like jail time.
That's right. That's right. Just for clarity, there was no jail yet involved. But I think that gives him a particularly unique view on the field, like this 30,000-foot view of all of the research they supported at DARPA, but then combined with his own research. I think it gives him a really unique and exciting perspective. So he's also a great guy. I'm going to turn this over to him. He's told me that he would entertain questions as they come from the audience, so please raise your hand in the middle. If you need something, I'll try to give you the microphone. Otherwise, I'm going to turn it over to Doug. Thanks for coming.
Thanks, Garrett. Thanks, everyone, for coming. Thanks to the people online for dialing in. Well, I guess you don't dial in anymore. You know what I mean. As Garrett mentioned, I'd be happy to take questions as I go through this. I know that this is a range of, the people here have a range of backgrounds from physiology to more hardcore engineering. So if something is unclear or you have a question, please don't hesitate to stop me. So my work really focuses at the intersection of engineering and neuroscience, and specifically how to create devices that can directly engage with parts of the nervous system to monitor and hopefully improve neurological function, particularly as it relates to sensory and motor functions. Just a couple of disclaimers. As Garrett mentioned, I'm very committed to doing translational work, and as a result, I work with a number of mainly startup companies, including one that my colleagues and I formed just a few months ago called Reach Neuro. So what got me into this space was really this recognition that, while modern medicine does enable some amazing things, there are millions of people that are living among us that have permanent disabilities that we can't really do much about. Someone has an amputation, that limb's not growing back, no Band-Aid is going to bring back that function. Same with spinal cord injury. Certainly occupational and physical therapy does amazing things for people that are recovering from spinal cord injury and even stroke, but once those patients sort of reach the chronic state of their recovery, things are kind of set. That is, their likelihood of gaining function beyond that sort of stable plateau is really low. And so in the absence of true cures for these and other conditions, we have to look for alternatives, to sort of compensate or correct for or minimize the burden of these devices or these diseases. And so, being an engineer and a big fan of robots, everyone loves robots. There are, I think, exciting demonstrations of what's possible with mechatronic limb systems. Right, and I'm showing a video here of the so-called DARPA arm that Johns Hopkins University Applied Physics Lab developed. It's arguably the most anthropomorphic robotic limb in the world. It has 22 degrees of freedom. It's got a hand that mimics much of the dexterity of the human hand, a fully articulated wrist, and as this video shows, it can do human-like tasks remarkably well. But obviously, for someone with an amputation or even a spinal cord injury to use a device like that, they need, essentially a high bandwidth connection from their body to the arm to get the arm to do the will of the user. And as well, when we use our native arms to interact with objects, it's not just the actions that the robot does, but the feedback that we get in performing those actions that is critical. So it's a two-way street. We need bidirectional communication between these anthropomorphic limb systems and the human body. And, maybe 10 years ago, it seemed like a big, bold idea, but now it's becoming very mainstream to build direct communication channels and communication systems that establish the two-way flow of information between devices, robotic limbs and other things, and the nervous system. And the reason this is possible is that engineers are now equipped with equations, largely developed by neuroscientists, that tell us what the map between neural signaling and sort of body state is, in many domains, whether it's the neural coding of tactile information that we get from our cutaneous mechanoreceptors, or how the brain represents actions that are being performed by the arm and hand. There's a neural code that isn't always perfect, but it gives us a starting point to interpret information that we measure from the body. And as well, we have physical devices that allow us to build these direct connections to different parts of the brain and spinal cord. Right? So we have a physical substrate, we have a way to connect to it, and we have the knowledge that allows us to read and write information with fairly high accuracy. So in the talk today, I'm going to focus on kind of two broad areas. First, starting with reading information directly from the nervous system. I'm going to start by talking about some old work and then some of our more recent work, where the goal is to be able to harvest information, harvest motor signals from different levels of the nervous system, interpret that information to predict what someone with a disability is trying to accomplish through their paralyzed limb. And we'll talk about intracranial and wearable sensors for doing that. And then in the second half of my talk, I'm going to focus on the reverse problem. How do we take information from the outside and send it back into the nervous system to not only enable touch and perception, but also to improve the regulation of motor output? Because as probably most of you know, the actions that we take, the movements that we make, and the actions of our muscles is conditioned critically on the sensory inflow that is present during performance of those actions. Right? So that sensory feedback plays a critical role in shaping motor output, and so providing or restoring those sensory feedback pathways, I think, is critical to achieving normal motor function. All right, so looking way back, so I did my PhD with Andy Schwartz and Jiping He at ASU, kind of in the very early days of multi-channel recording in motor cortex of a variety of animals, but primarily rhesus macaques. And Andy's work with Apostolos Georgopoulos and others, back in the 80s, found that there's this very nice kind of stereotyped relationship between the reaching movements that an animal makes with its arm and the pattern of spiking activity that you measure if you poke around motor cortex and look at neural activity. That is, neurons in motor cortex, or at least many of the neurons in motor cortex, exhibit what's called a preferred direction, where their activity is maximal for movements in one direction and gets progressively weaker for movements away from that preferred direction. And they further went on to show that if you take the collective activity of a bunch of these so-called directionally tuned cells and you put it together, you can actually predict the actions that the monkey's going to make 120 milliseconds before the arm actually moves, right? So there's this movement predictor or movement indicator signal that you can generate if you sample from enough activity and run it through the right sort of transformation. So that really got us started on this path of building real-time recording and decoding interfaces that would allow us to extract movement command signals in real time, channel them out to a robotic arm as shown here, and then eventually enable the monkey to use the arm through direct thought control. So I'm going to try to play this video now. So this is from Andy's lab after he moved to Pitt in 2002, I think. And here, the monkey has arrays of electrodes implanted in primary motor cortex. It's hidden behind this 80-20 frame here. When he leans forward, you can see the cables. But the signals are being read from 100 electrodes implanted in motor cortex and then run through this population vector decoding algorithm to drive the endpoint velocity of this hand, right? So this arm has 3 degrees of freedom to control the position of the gripper, and then a 4th degree of freedom to control the opening and closing of the gripper. And as you can see, the monkey's able to move the arm to any arbitrary location in space. Doesn't matter where the marshmallow is, as long as the arm can reach it, the monkey can direct the arm to the target and close the gripper to retrieve the marshmallow, with pretty high accuracy. You may have noticed that he does make some mistakes. He pulls before having a firm grip, and he's able to go back and course correct. Right, so it's in many ways it looks like a very natural task that the monkey is able to perform with, I think, some pretty impressive agility. Okay, so fast forward then a couple of years. So this really sort of laid the foundation for building devices that we thought humans could use to perform activities of daily living after experiencing a severe injury that causes paralysis, such as spinal cord injury. So working with folks in the medical center at UPMC and Jen Collinger is shown here. Jen is a professor of PM&R at University of Pittsburgh and an early leader of this project. This photo shows our first participant that joined the study in 2010, I think, 2010, 2011. She was a 52-year-old female at the time and was essentially paralyzed from the neck down due to a condition called spinocerebellar degeneration. So she was unable to move any of her limbs and read about our study and was eager to participate. And so she signed up and she received a pair of these 100-channel Utah electrode arrays that were implanted into the arm and hand areas of her motor cortex, which we mapped out using functional neuroimaging. So that told us where to place the arrays. And then once the electrodes were in position and we mapped out the relationship between neural spiking and the movements and her intended actions, Jan was able to perform a variety of tasks that involved controlling that robotic arm to reach, grasp, and manipulate objects in her surrounding environment. And so now I'll just show a video, which I think is the best way to demonstrate some of the things that Jan was capable of doing using this direct brain interface. So there you can see the connectors. So the thing that I think is most amazing about, what Jan was able to do is that it's completely free form, unscripted. It's not like we gave her a set of 5 tasks and said, you can do these 5 things, and we're going to use machine learning to kind of classify your neural activity into those discrete bins. This is a continuous, like the decoding methods that we use provide this continuous map between her neural activity space and her kind of kinematic state or workspace, right? So she can essentially perform any gesture that she wants within the reachable workspace. And she's able to do that quite effectively using this approach, right? So it's almost effortless. It seems like it's almost effortless for her because of the great flexibility that the decoding approach, I think, provides. So that's, I think, an exciting demonstration of what's possible, obviously in humans, but there still are some challenges that we need to overcome to really sort of bring this into the mainstream. This is technology that is still relying on connections that have to go through the skin. The wires have to come out through the skull. There's a connector that's attached to the skull and runs through the skin. So that's not yet in a form where it is suitable for, I would argue, true mainstream clinical practice. It takes a team of researchers to sort of maintain these implants, but still, a very promising indicator of what's possible. Maybe Elon Musk and team will do a better job of getting rid of some of the connection issues. Anyways, exciting potential for intracortical BCIs. We're also interested in identifying potential technology paths that might have a lower barrier to adoption, right? So if there are devices that can be worn more like garments that we all put on every day, most of us put on every day, before we go out into the world, if those garments could somehow be sensorized to detect the types of activity, the types of signals we need to infer someone's intent to act, then that could become the interface. And I couldn't imagine a more less obtrusive way to reconnect humans with their environment. So we started working on this concept a few years ago. The main student on this project is Jordan Ting, her picture is shown at the top right, and Alessandro Del Vecchio, who was at ICL, Imperial College at the time. He's moved on to a faculty position in Germany. But the study that we did involved this high density electrode array that was embedded into the lining of a sleeve that you can see here. Let me go back one slide. So this sleeve has an array of simple disc electrodes that were designed initially for electrical stimulation, with the idea that we could electrically stimulate muscles throughout the arm. But we realized that, with that direct contact with the limb, we could also measure EMG activity as well. And so the idea is we strap this sleeve onto the arms of patients, and in this case, these are 2 patients that have complete spinal cord injuries and no function in their hands, and they're both participants in BCI studies. We thought, maybe with an electrode array that's that big, that provides that broad of coverage, that maybe we could detect very small levels of muscle activity that might be persistent after their spinal cord injury. And so we it was like looking for a needle in a haystack, but we had a very large electrode array to help improve our chances of finding something. And so here's just a short video showing Ian attempting to perform a set of gestures that are cued on the computer screen. So here he's being cued to do grasp. You can see his hand is not moving because he's unable to open or close his fingers. But when he's given the cue, we see clear but weak patterns of activity in the muscles. Right, so the muscles are there. They're not strong enough to produce any action in the hand, but they're strong enough to express myoelectric signatures of that intended action. And so Jordan and Alessandro worked together to take that array of high-density EMG activity and apply a technique called motor unit decomposition, or which is essentially a blind source separation process that is able to resolve the single motor unit action potentials that give rise to the interference EMG signal that we're all familiar with. That is, we can deconstruct the components of this, the source components of this signal if we have enough electrodes. And so this just shows some examples of the spiking activity that we're able to resolve after applying that process. And then if we look at the pattern of spiking activity that we observe as these subjects are attempting to make these movements, we can see that there's clear, like in this case, the task is wrist flexion and extension, and we see clear alternating patterns of activity where flexor motor neurons are active during the flexion phase, extensor motor neurons are active during the extension phase, and we see this nice rhythmic alternation. And in this case, the subject's wrist is weak. It's certainly not fully paralyzed, so it wasn't surprising that we could detect this level of kind of task-specific activity for the wrist. If we apply non-negative matrix factorization, we can compress these 7 motor units down to 2, and we see nice kind of rhythmic alternations of that extension and flexion activity. But the story looks a little bit muddier when we go to a digit where there is no movement, right? So in this case, this is for his index finger, and so now that the subject is being asked to flex and extend each of his digits individually, and he's unable to do so, right? He's trying, and he's given a cue that tells him when to flex and when to extend, but he has no sort of feedback or any indicator that he's doing the task correctly. But in spite of that, we still see activity, we still see spiking activity in groups of motor units that we can resolve from this high-density EMG array. And again, if we apply NMF to the firing rates that we generate from these patterns of spikes, we see that, if you squint really hard, you can kind of see it looks like there's alternating activity, at least on the extensor side, which we know is a little bit stronger based on the deficits at the wrist. But the alternating activity looks to be a bit weaker, if present at all, on the flexor side. So, the point of this is to show that, in a digit or in a set of muscles that are, apparently fully paralyzed, or at least functionally fully paralyzed, we are seeing voluntary activity expressed in the pattern of motor unit output. And one, I think, maybe clearer way to show that is to compute the power spectrum for those NMF signals, where at the wrist we see a strong spike in the modulating power at the movement frequency of 0.5 hertz. Right? So this tells us that the subject was deliberately modulating output to those muscles in time with the task. And while we knew that was the case for the wrist, because the wrist was actually moving, at the index finger, where there was no action to observe, no physical movement to observe, we also see that, in fact, that voluntary modulation in motor output is present. Right? And I think this is really important, right, because this tells us that even though these are people with quote-unquote complete spinal cord injuries, they do retain some ability to voluntarily control output to muscles that are too weak to do anything sort of in the physical world, but might be strong enough to express activity in sort of a digital world, if that makes sense. Yeah, Garrett.
In the community might be wondering this also. I'm wondering, as if you take the sensors off, put them back on, or just from day to day, from subject to subject, can you say anything about how well that maps down to some kind of invariant signals that would reflect the movement?
It's an excellent question. It's something that it is a problem that will have to be solved at some point by someone in the future. If this is going to be, if this garment idea comes to life, then when you put your shirt on in the morning, it's going to have to calibrate to your body state. I think that's a solvable problem. I can imagine a couple of ways to at least attempt it. But yeah, we're not far enough along to comment on that, but it's definitely one of the challenges on the list.
Do you want to go ahead and unmute yourself and ask your question?
Oh, sure. Hey, really neat stuff. I was wondering, do you see spasticity or other non-voluntary movements sort of creating a pattern in this high-density EMG for people with?
So, excellent question. We do see spasticity, which at present we are ignoring because it's an inconvenient. So that's an involuntary signal that that presumably doesn't tell us anything about what the subject is trying to do, or may not tell us anything about what the subject is trying to do. So it's a good point. It's something we will likely have to deal with, given the features of that spastic activity. It tends to be synchronous and large in amplitude, and so I think discriminating a spastic bout of activity should be straightforward relative to the more voluntary outputs that we're seeing here. But
yeah,
another one of these.
The reason I ask, is in terms of tracking spontaneous recovery or something, perhaps that could be an indicator of improvement.
No, that's an really excellent point. And that is, I think, one of the other potential, maybe valuable use cases here is that we think this provides a very comprehensive look at what's going on at the level of motor neurons in the arm. And so if you have a patient going through a therapy that is intended to, restore normal motor function in the arm, this, I think, would provide a great deal more insight into what's going on under the hood, so to speak. All right, let me move on. So, so I showed in the previous slide that there's what we are interpreting as evidence of clear voluntary output to muscles that are paralyzed. This slide shows that if you look at the population activity, so if you take all of that activity and you sort of compress it down using principal components analysis, and then look for differences in the population activity across tasks, that we see that there's nice, fairly clear separation. Even when we use EMG power, there's, it's a little bit hard to separate for some of the joints. But if we look at the neural activity that's expressed by the motor unit firing rates for each of the 5 digits that we're asking the subjects to imagine or attempt moving, we see nice, very clear separation. There's very little activity for the thumb, so that makes it a bit of a standout. But the other 4 digits show nice, strong patterns of modulation that seem to occupy their own part of this low-dimensional space, which is, I think, good. And then if we apply classification methods to this data, we see that the motor unit spiking is, in general, more discriminable than the EMG power. There's certainly less confusion with other joints, and average classification accuracies are a bit higher. Yeah?
Yeah. Question about, so here you're either using, so you're checking out the phase when you're looking at EMG power, and then you have this motor unit decomposition.
Mm-hmm.
So I imagine there's something in between where you still keep the EMG waveforms, but you're not doing the motor unit decomposition. So if you just do PCA on the raw waveforms, how does that look?
On the EMG waveforms?
Yeah, yeah.
So we haven't done that. So here what we're doing is we're taking the spiking data from here, running it through a low-pass filter, essentially, to get a smooth firing rate. So we have an envelope that looks something like an EMG envelope, right? If you take
Yep.
raw EMG, right? And so then in both cases, we took that data and reduced it down to 10 principal components and decoded on that. And the reason we did that is we wanted to make the comparison a fair one, right? Because we don't have as many motor units as we do EMG channels. That makes sense.
I see. Yeah, so I'm wondering, so is the motor unit decomposition linear?
In that, if you added up all of the action potentials, yes, it should be.
Interesting. Yeah, so I'm just wondering if there's some information that's being left on the table, in between doing the motor unit, decoder and the EMG power decoder.
Well, so my interpretation of it is, but if that were the case, then the motor unit decoding would be lower, right? Because you're saying that we'd be losing information, and I'm saying that it's actually more specific. So I think of it more of a denoising step, like the raw EMG, even though there's more channels, also has more noise because things are more blurred together.
Gotcha. Okay, cool. Thank you.
But, okay. So bottom line is we think it's voluntary like it's voluntary motor output, and it discriminates well for attempted actions. What we haven't done yet, and I think it's going to be an important next step, is to actually do this in real time, and give the subjects actual feedback of what, to show them that, look, even though your finger's not moving, you're starting to produce muscle activity that is task relevant. And I think, I hope that in doing that, it will reinforce the activity to be even stronger. All right, so now let's talk about the other, taking information and sending it the other way. So I'm going to move quickly through the first. How much time do I have, Garrett? 12:15. Oh, okay, so then I'm okay. So I made this point earlier, but I want to restate it, that we think about motor control, and especially the people that study cortical motor control, have this very sort of top-down view of how actions are controlled by the nervous system. Motor neurons in primary motor cortex sort of are the be-all and the end-all for dictating actions. And the reality is, much of the activity that we measure in our muscles is driven, not just by input from descending fiber tracts, but also from recurrent feedback that's coming from the limbs themselves. That is, sensory feedback from the limbs is critical in shaping the actions of the muscles. And so, and if we think about that and the impact that has on the BCI work that we do, a lot of the BCI work is really trying to sort of kind of restore kind of normal motor control by bypassing parts of the circuit that are not working or that are damaged. But we're not sort of bypassing all of the critical pathways when we do it, right? Because, subjects can see the actions that they're performing on a screen, but they're not feeling them the way that they would if their limb was actually engaged in the task. So all that to say, I think restoring somatosensory feedback is a critical part of building a true bidirectional sensory motor interface. All right, so if our goal is to restore inputs to these somatosensory pathways, where can we go? The answer is there's kind of a lot of places to choose. Folks here, Garrett and Chris, I know, have been looking at stimulating in primary somatosensory cortex, so you can start up high and pattern information there. Most of my work is focused on peripheral nerve and spinal root interfaces, so down here at the level of the dorsoroot ganglia and spinal cord. And we did a lot of early work in cats to sort of sort out where to go and what to stimulate. But more recently, we've shifted to using humans to test the effects of sensory stimulation on action. And so let me tell you a little bit about that. So epidural spinal cord stimulation is a technique that's used quite commonly for treating people with pain syndromes. It involves electrodes, usually one or more linear or paddle-style electrode arrays that are placed just on the dorsal surface of the spinal cord. And so they rest kind of right on top of the dura. And depending on where they're positioned, they tend to stimulate the large diameter sensory fibers that are projecting into the spinal cord from the peripheral nerves. That is, we're stimulating, or we have the ability to directly stimulate the primary afferent pathways that are carrying information from muscles, skin, and so on. Okay, and so doing this in humans gives us the ability to, having technology that is approved for human use gives us the ability to test this technology in patients where we want to ask questions like, what does that feel like? Do you have phantom limb sensations, and how are they changing with stimulation? Okay, so the first project that I'm going to describe is a project that involves using spinal cord stimulation to try to restore sensory input to amputees, specifically transtibial amputees. This is a project led by Lee Fisher, one of my colleagues at the University of Pittsburgh. And the approach is to take up to 3 8-channel spinal cord stimulation leads and implant them into the lumbar, primarily lumbar spinal cord, to try to stimulate the sensory dermatomes that are associated with the lower leg. Right, so we're recruiting transtibial amputees, and we'd like to be able to bring back sensations that are referred to that part of the leg which is no longer there. Here's an X-ray showing what one of the implants, what a pair of implants looks like right after implantation. These leads are placed on a temporary basis, right? So they're spaghetti-like, so they can be threaded through a catheter into the epidural space without having to do an open surgery, and so then the lead can just remain in that that percutaneous form for up to 90 days before they're retrieved. And so it gives us a kind of a nice window for testing the ability to bring back these sensations and measure their effects on the actions that the person does. This is not yet the sort of long-term solution for a sensory restoration device. Ultimately, we want to have a fully implantable system that has no wires going through the skin. All right, so this shows the patient's own drawings of the types of sensations that they experience when we electrically stimulate different contacts on those arrays of electrodes. And so you can see that in some cases they feel sensations that are distributed broadly across the entire leg. In other cases, they feel sensations that are referred to just a few toes, and so on. But I think it's important that in many cases we're able to produce sensations that are associated with the part of their limb that's missing. And one thing that's still a little bit peculiar, and we're still sorting out, but it seems that time post-injury may have an effect on their ability to actually feel sensations in the limb. So if you take someone that has just had an amputation, and again, this is anecdotal at the moment, but it seems like they are more capable of feeling sensations in their missing limb than someone that's several years out. That it's almost like they have to kind of relearn or sort of rebuild an awareness of that part of their body. Even if they have phantom limb sensations, even if they experience phantom limb pains, it's like they almost forget how to interpret sensory information that's coming in from the periphery. But I don't want to say too much about that because we still don't really understand it, but it's interesting. One thing that we are growing ever more confident about, though, is that when we do sensory stimulation for, well, now we go out beyond just a month, we're out to 3 months, we see that there's a progressive drop in the phantom or in their pain scores that they report over the course of the study. So phantom limb pain is a very common symptom in these patients, and restoring sensory feedback, even very crude sensory feedback like we provide in this approach, does seem to help. And so I think that's definitely encouraging. I also want to show us just some quick videos showing the effects, the effects that we think we're having on the ability to ambulate safely and effectively. So we built a closed loop system that takes sensor data from the foot, turns that force data or pressure data into a pattern of stimulation that is updated on the fly as the subject walks. And then we test their ability to walk in this narrowing beam walking test, where the subject is essentially walking on a balancing beam that gets progressively harder as they progress along the length of the beam. And the further they progress, that is, the more narrow the base of support that they can stand on, presumably the better they are at, the more stable is their walking. So let me just play this video now. So this is our most recent subject. On the left, she's walking without stimulation. On the right, she's walking with stimulation. I'm just showing a single trial, but at least in this case, she does seem to be able to walk further or reach a longer distance along the walkway than when she doesn't have that sensation from her foot. So we think that there's some functional benefits. We're also interested in, and we have been spending a lot of time focusing directly on the effects that the stimulation has on their motor output. I tried, I think we believe that providing this sensory feedback should improve stability, but it could also mess things up as well, because these sensory inputs can have powerful effects on motor output that may not always align with the task being performed. So we're using EMG to survey the responses in muscles. So Dev Sarma is the postdoc on this study. We're surveying the responses in muscles as we apply stimulation at different levels of the spinal cord. We see what we know are reflex responses in those muscles, because if we do a rate-dependent stimulation test, we see clear suppression of the responses for second and later pulses. So we know that we're activating muscles transsynaptically. The question is, can we produce activation of those muscles in a way that would be synergistic with the actions that the subject is trying to do? And so here I'm just going to show you a couple examples of EMG that's recorded as the subject is walking without stimulation on the left. We see nice modulation in hamstrings. We see a little bit of modulation in the gastrocs, even though the gastrocs isn't really being used here because the subject is a transtibial amputee. We see very little activity in this case in vastus lateralis. But when we apply stimulation, and so now the subject is walking and she's getting sensory feedback from her foot, we see, I think, what looks like clearer phasic modulation of the gastrocs muscle, which again is still sort of functionally useless. It's not driving. There's no ankle for it to drive. Maybe it has having some effect on the knee. But we see nice, stronger bursts of activity in gastrocs, and I think very strong bursts of activity now in vastus lateralis. So, is there a question?
There's a question from the virtual audience from someone named Omer Inan. This person asked, how is the sensor data from the pressure insoles processed and fed back to the stimulator? And is it asymmetry that's captured, for example?
That's a good question. We didn't measure symmetry yet, although we certainly could look at the kinematics and do that. The algorithm for converting the pressure data to spiking rates, there's sort of a library of algorithms that we're using. Some are based, it's just a linear map of pulse rate to pressure. That didn't actually work that doesn't seem to work very well. So we're using a new algorithm that is pressure plus the time derivative of pressure. And so there's the combination of those two things that drives amplitude and firing rate. I don't know which specific algorithm was used here, though. It's a good question. They're all simple, I think, is the important point, though. All right, so again, one of the things that I think is really important is that stimulating sensory inputs to the spinal cord does provide a path for driving activity in muscles whose function is compromised by something, whether it's stroke, spinal cord injury, amputation. And so we thought, or we're looking to extend that then to other conditions where muscle function is compromised. And so certainly people that experience a stroke, typically experience deficits on at least half of their body. It's, upper limb and lower limb. For many patients, they recover a lot of that function within the first 6 months post-stroke, and so they actually do well. But for about half of the roughly 800,000 patients that suffer a stroke each year, their deficits are permanent. And so there's really little we can do when someone reaches the chronic phase of their recovery, because therapy is largely ineffective at that point. And Botox and other things work for spasticity, but don't really add function, in my opinion. So we thought, well, maybe spinal cord stimulation could provide benefits similar to the way that functional electrical stimulation provides benefits, but perhaps in a form that is better for the patients. And so this is working with Marco Capogrosso, who's a new PI at the University of Pittsburgh in the neurosurgery department. His postdoc, Mark Powell, who is a GTech alum, he wanted me to say, and joined our group about a year ago after doing his PhD at Brown University, and Nikhil Verma. There's several other students on this project as well, but I'll be talking mainly about their work here. So the idea is that if we can stimulate sensory inputs in the cervical spine, that we might facilitate inputs that are driving and coordinating activity across muscles that span the shoulder, elbow, wrist, and hand. And so this is roughly what the map looks like, or the segmental organization of those motor neuron pools looks like in human. And so we designed a study where we're implanting arrays of electrodes that span roughly C3 to T1. So this X-ray shows that here. There's a pair of 8-channel leads that are implanted in the cervical space. The lowest one is at roughly T1, and the highest one is up here at C3. We've done 3 30-day implants so far. The first one was in May of last year, and our first patient was 9 years post-stroke. So she was almost a decade without the use of her left hand. Her level of impairment was moderate. Our second patient was severe. So these are people that are really compromised in terms of their motor function. And so we thought, maybe that this approach would offer some benefit. I'm just going to show you a couple of recruitment maps. So when we do these implants, the first thing we do is try to find which muscles are responding to stimulation that's delivered at different locations. And so it's sort of an anatomical search, really. And so the rostralmost contact in this case activated muscles at the shoulder and in the upper arm, and the caudalmost contact on this rostral lead gave us nice activation in the forearm in this patient. Okay, and if we look across all of the contacts, we see that we can produce activation in different groups of muscles. It's never a single muscle that's responding to stimulation. We're always getting groups of muscles in that are responding to stimulation, but we find functional groups that are useful. And so we're doing a variety of tests to assess the impact of stimulation on function. One of the things we do is use a dynamometer to measure joint strength, so just single joint movements. And so here are some torque and EMG plots for our first 2 patients. We see in both cases the torque output and EMG output in the presence of tonic spinal cord stimulation is much greater, so we're facilitating a higher level of motor output at those joints. At the shoulder as well, we see nice, strong increases in torque output in the stimulation on condition. Interestingly, it's a little bit more tricky to measure EMG at the shoulder because it's closer enough to the site of stimulation that we're dealing with a lot of artifact. We think we've solved it, but I'm only showing the torque outputs here. So stimulation definitely boosts strength, even in muscles that are really, really weak, as the case of SCSO2. Now I'm going to play some videos that show, I think, provide a nice, clear demonstration of the types of effects that stimulation has on function. So here we're just looking for range of motion. So in this case, this is the very first day of testing with this patient. So she hadn't had any spinal cord stimulation before, and she's 9 years post-stroke. And so without stimulation, she really struggles to open her hand, and she can open it maybe just a little bit. With stimulation on, she can extend her digits fully, to fully open the hand. You'll see, I think, in a later part of the video, she's able to grasp, a soup can because of that increased range of motion. As well, in different contexts, enhance function more proximally. So here, without stimulation, she's unable to reach above her head, which is obviously an important activity for getting dressed in the morning. And without stimulation, she really struggles. With stimulation, it appears like it's much more effortless for her to do. If we look at dexterity, so those I think are some nice demonstrations of the strength improvements. We also see improvements in her ability to coordinate action, especially at the hand. So here she's doing the box and blocks test without stimulation at the top and with stimulation on at the bottom. And she fumbles quite a lot without stimulation. The hand, I think, is just, in her words, it's just not very responsive to what she's trying to do. And when stimulation is on, she feels like the limb is just doing what she's wanting it to do, like it's lighter and more responsive. So we don't have to watch that. It's kind of a boring task. But the bottom line is she's able to complete many more transfers when she has the assistance that's provided by stimulation. And then lastly, just some demonstrations of the impact that stimulation is having on her ability to perform activities of daily living. So here she was asked to do a task that simulated eating. I think this is the actual steak. So for the last day of testing, we actually went out and got steak for her to eat. Before this, we just had Play-Doh that was sort of shaped like steak to test her ability to cut and grab items of food off of the plate. And you can see without stimulation, she has a really tough time holding the fork. She has to relies heavily on compensatory movements to complete the task. But it's a lot of effort. And as a result, patients, tend to not use the arm, right? Because it's just a heck of a lot. Well, not for everybody, but for many people, it's just easier to sort of live life with one good arm. Here, she has stimulation turned on. She's able to pick up the fork. She still has to rely on her other hand, but now she's strong enough to hold the fork with a grip that's strong enough to keep it in her hand and not slide out. And her elbow and shoulder are strong and sort of limber enough to allow her to actually bring her hand to her face. And the thing that is, I think, worth noting here is that this is just tonic stimulation. So there's a couple of contacts that we're activating in the spinal cord, but there's no phasic modulation of that activity in this task. They do a lot better if we actually dynamically control the stimulation output. But even with the simplest mode of stimulation control, they do pretty well. Some of them do really well. Patients with more severe impairments don't respond quite as well. Anyways, just the last thing I want to show is that if we look at impairment scores measured by the Fugl-Meyer, we're impressed that we're seeing what look like significant changes in their motor function over the course of the study. So the first patient saw a 12-point improvement in her Fugl-Meyer. The second patient saw only a 3-point improvement, but that's a lot for someone with that level of impairment. We didn't expect this. We were expecting that the benefits would be afforded only when the stimulation is active. This is showing that there's actually some carryover effect, that, you know, in just 4 weeks of testing, their arms are getting better. Maybe, you know, maybe not enough to make a difference, but, you know, the fact that we're not even like trying to help them get better, I think suggests that there's a lot of opportunity to make this a more effective therapy if that's the goal. And then as well, the Ashworth scores are shown here to make the point that we don't think we're exacerbating their spasticity, which is something we were unsure about when we started, because spasticity is often associated with hyperreflexia, and the fact that we're directly stimulating reflex pathways to get these effects made me at least worried that that we might worsen their spasticity. That doesn't seem to be the case. Question?
Yeah, Lucas McKay had a quick question about the video that you showed. He wanted to know whether there is a sham stimulation condition that you used for it, and also whether or not she was left-handed for eating that steak to begin with.
I think she was right-handed. It's a very good question, but I think the fact that she hasn't used her left hand in 9 years is probably the best. Well, that and the fact that she has a stroke. But yeah, no, we do have a sham. I don't have any of the data from the sham condition, but there's a clear difference. So it's hard to do shams because they can feel the sensation. So we had to come up with a stimulation pattern that they could feel and not discriminate from one that was, you know, the effective mode of stimulation. But anyways, all right. So I'm sorry I went way too long, but I wanted to, you know, talk about those distinct areas of the work we're doing. I'm really excited to see so much of the neurotechnology work moving into humans. You know, we're just one of many groups that are now working very aggressively to take new technology and try to get it to help people. I think Chetan just joined the BrainGate team, right? So there's going to be, I think, more and more of that work happening here. Chris has been involved in a lot of translational neurotechnology projects. So I think it's super exciting because I think these devices have real potential to help lots of people. So, thank you.
Okay, we do have some time for questions, so I'll hand the microphone over as soon as I see a hand. Anyone? I'll start just to get people, break the ice a little bit. In the example of the stroke patient where you showed her moving the blocks, I thought on the right side was an image of her other hand. Was I interpreting that? And she seemed to be doing something with her other hand. I'm wondering.
You see that in a lot of the videos. So a lot of stroke patients will make mirror movements when they're trying to use their impaired hand. It's something we actually want to try to quantify. And if you look at the videos, you see stronger mirror movements without stimulation than with stimulation. But yes, it's a real thing.
It made me think.
It's a strategy for trying to boost output to the impaired side.
Yeah, it made me think of, we recently had a seminar speaker who was talking about a robotic hand for amputees, and they actually would show the individual, the mirror.
That's mirror box therapy, probably.
Yeah.
Yeah, so that's different. So there you're trying to sort of fool the brain into thinking that your amputated limb is intact, right? Because you're trying to reinforce the body schema that, you know, that body part is there. In the case of mirror movements in stroke, and you know, I don't know how well established this is, but my understanding of it is that you're relying on ipsilateral connections, right? Because the dominant source of corticospinal input, you know, crosses over, right? And so, you know, and that's, you know, in her case, that's the pathway that is damaged. There's a small percentage of ipsilateral fibers, I guess it would be ipsilateral, would be her left hemisphere, that can facilitate output as well. And so it's, you know, the thinking is that when you see someone making mirroring actions on their their healthy, the healthy side of their body, that they're trying to drive those ipsilateral pathways.
Got it. Yeah. Okay. Thank you.
Right.
That's very interesting. There's
Yeah,
so many
super-
questions behind that. So, Ming, you had something? Okay.
And so this one is from Stephen Wolf. He wanted to know.
Steve Wolf.
Yep,
Nice.
pertaining to the second part of your talk on EMG. So he asked, how can one record EMG from a deep muscle, such as a finger flexor, with surface EMG? Will those activations be recruited reliably? And the patient whose data you showed may be quadriparetic and not plegic, and perhaps the clinical diagnosis was inaccurate and the cord lesion is not complete.
I think there's a lot in there. I'll start with the last question. So there's a term which is new to me, but I don't know how long it's been in the literature, called discomplete, where at least in the case of spinal cord injury, when patients are given their initial diagnosis as motor complete, that is based on their ability to generate action, force at different joints, fingers, wrist, elbow, and so on. And this subject's manual muscle test scores are all 0 for all digits of the fingers, all 5 of the digits. Wrist is 0 for flexion and like 1 for extension. It's severely impaired, and then it gets better as you go up. So for sure, it depends on what you define as complete, right? Is there a complete loss of motor function at the hand? Yes. Is there a complete transection of the spinal cord that prevents all descending input to go to the motor neurons? Obviously, no. So, anyways, it's there's a bit of semantics, I suppose, in the literature about.
There's another question over
here.
Yep.
Yeah, kind of a more generic question, but I think piggybacking on Garrett's point, which is I think there's been a dialogue about this in the small animal community, but in the context of brain-machine interfaces, either peripheral or centrally controlled, do you have a sense for sort of how specific the control is? So whether it kind of starts as very generic, make any movement that creates a signal for the interface to pick up on, and then it becomes more specific. And the reason that I thought about this was some of the early videos you showed of the monkey. It was pretty clear there were like a bunch of movements, right? Not necessarily mirror movements, but they're kind of wiggling their hand,
Yeah.
right? And so do you have a sense for whether this might be different in a centrally controlled brain-machine interface versus one that's peripherally controlled?
I can only speculate that a peripherally controlled interface would be a bit less flexible than a centrally controlled interface. It's difficult to coordinate certain types of actions, at least at the level of digits. I don't know that anyone has demonstrated that you're more dexterous with a cortical interface, but I can imagine that it would be a bit more pliable. I think it really depends on the task itself, how intuitive is the task, and the map that you're using to connect whatever signal you have to the task. Because your ability to link task performance to some change in your strategy is critical, right? Because if you see a mistake but you have no idea how to correct it, you're never going to get it to work. Whereas if it's if the errors are interpretable, then it's much better.
Maybe I'll just end with one more prerogative of the microphone holder. And this is asking you to kind of speculate a little bit, but with this whole arc for the neuroprostheses, where's the biggest bottleneck or sore spot for you in terms? Is it hardware? Is it algorithmic? Is it, if you had to really had your wish of one area to really emphasize?
So thinking only about technology limitations, is that?
No, I think you take it how.
I think hardware-wise, we're pretty. So first of all, there's a big difference between the hardware that we use in the animal labs and that we use for a lot of our fundamental research and the stuff that we're thinking about using in patients, right? We have 100-channel Utah arrays that we use in our animals and even our humans, I suppose. Whereas, for humans, maybe a dozen channels is all you're going to have to deal with. Just there's a simplicity to human-grade devices that is essential. Although maybe Elon Musk will change that with his Neuralink system. I mean, I think the answer to your question really depends on the application space. For BCI, I think that there are simple activities that are going to be simple functions that are going to be high value. Digital communication is going to be high value for a lot of people. Allowing them to interact with their computers if they can't, because their hands are not functional. And algorithmically, I think there's definitely opportunity, and Chetan has done a lot of work in this space on ensuring stability over, an expanded time scale, because the device has to be intuitive, simple, easy to use, or at least seem to be simple, intuitive, and easy to use, even if under the hood it's, powered by some crazy, complicated deep learning system. So, I think there's certainly a lot of space.
Yeah, okay. All right, so that's great. I think it speaks to a lot of our community, different people working on different pieces of that. So let's thank Doug again.